नमस्ते तिवारी जी।
नमस्ते सर, गुड इवनिंग सर।
गुड इवनिंग, गुड इवनिंग। काफी मीटिंग हो रहा है ना, इसलिए जरा टाइम नहीं मिलता है।
थैंक यू सो मच सर फॉर ज्वाइनिंग।
एमबीएस स्टूडेंट्स को भी इंस्पायर करना है ना हमको,
हम्म।
उसकी वजह से। और मेरे जो दो स्टूडेंट है, दे आर वर्किंग ऑन पोस्टबायोटिक। आई टोल्ड देम आल्सो टू
ज्वाइन सर।
come here online, so we can take them their opinion also. Anjali Jaglan is there, she can introduce.
ओके, सी इज हियर सर, अंजलि जागरण।
Good evening, sir. Hello, sir.
So good evening, respected faculty members, scientists, panelists, and all the participants, and my dear students. It is pleasure to welcome you all in the today's roundtable discussion on beyond probiotics, postbiotic as future therapeutic agents. So all of us are very familiar with the terms probiotics, prebiotics, and the synbiotics. And over the year, these three terms become an integral part of the gut microbiome research or the healthcare and even now in our daily life. So these probiotics have been widely explored for their role in improving the gut health and the immunity. Prebiotics, you all know that support the growth of the beneficial microbes, and the synbiotics mainly the combined both for the enhance the effect of. So despite the popularity and the advantages of the probiotics, these probiotics comes with the certain limitation. So being the live organisms depends on the viability of the microbes, the conditions and survival. The most important one is the survival within the host. So there are also the septic concern in the certain populations, along with the variability in the or the differences in the clinical outcomes based on the different host. So now these all limitation brings to an emerging and the promising concept that is the postbiotic. So this topic is suggested by our Dr. Prakash Salami sir because he's working on this field and he's an out scientist in the postbiotics. So postbiotics, we all know that the non-living microbial cells and or they're the bioactive components that provide the health benefit to the host. So now, by using this term, the postbiotics, instead of relying on only on the live bacteria, we are now trying to focus on the functional molecules they produce. Okay? And those molecules may be the metabolites, enzymes, peptides, and any other the cell component. So now this shift from the probiotic prebiotic and symbiotics to the postbiotic, obviously this will offer the better safety and improved stability and more predictable effect on the host. And so this today discussion provides us an opportunity to explore these aspects, share the research insights, and to understand that how the postbiotics concept the future of the therapeutic. So now as we move beyond the probiotics, so obviously we will not replace the existing approaches, but we will try to advance towards more precise and effective solution. So once again, I welcome you all in this roundtable discussion, and I request all of you to share your research experience, to share your knowledge to all the participants present in this session. So now I request anyone, if you are interested to share your research experience or your knowledge in this postbiotic, you are free to unmute yourself, and if possible, you can open your camera also. Hello. So request everyone that if you want to share, just unmute and share your knowledge or the research experience with everyone. Let me, Anjali Jaglan.
Yes,
You are
good
watching.
evening, sir. Good evening, sir. How are
Anjali,
you,
just
sir?
fine. Just introduce yourself first so that everyone can.
Okay. Sir, my name is I'm Dr. Anjali Jaglan. Currently, I'm assistant professor in Delhi University.
It's
Co-
good.
uh, my, my college is Shahid Rajguru College of Applied Sciences for Women, and I have worked in the field of probiotics during my PhD. In which I have worked on celiac disease and probiotics, which are gluten degrading probiotics. So I have isolated them. So during my PhD, I have worked on not on postbiotics, but I have done one experiment in which we have seen the antimicrobial effects of the supernatant after like growing the probiotics. We have collected the supernatant and we have seen its antimicrobial property against different pathogens available in our lab. So I have done that. So in that we were not having any live cell, but supernatant of the probiotic. So I think that was postbiotics. We have not further studied like which postbiotic was there, and further we have not evaluated it. But yes, we have done this one.
Thank you so much, Anjali. Dr. Anjali.
Thank you so much, sir.
Sharing. Thank you. So now we have Professor R. Bharat Kumar, Dr. Renu Agrawal. Just unmute and introduce yourself first.
Tejaswini
became
there.
Pardon, sir?
Tejaswini.
Yes, sir. Hello.
Yes, Tejas.
Good evening, sir.
Good
Followed
evening.
by
I'm
Lekha.
currently working on. Uh, Good evening, sir. I'm Tejaswini. Currently, I'm a PhD scholar. I'm working under Dr. Prakash Lamy, sir. And currently, I'm working on studies of probiotics that influence on beta carotene. I'm targeting the short chain fatty acids, especially the butyrate, because I'll be working on colon part. So I was just looking into the in vitro fermentation and colonic to get that short chain fatty acids, and then seeing the bioavailability of retinoids and beta carotene and vitamin D homeostasis and so on. Sir.
Okay, thank you, Tejaswini.
Yeah, thank you, sir.
Dr. Malini Madam is also joined today. Am I audible to you, Dr. Malini Madam?
Yes, I am. Yes, you are audible. Good evening. And
Good
I
evening, madam.
joined, yes, I joined like especially for the topic. And again, since sir is there, Rami sir is there, like he just really did a lot for us. And so I would be happy to hear from him and
Good
his team
evening. Good
also.
evening.
Good evening. Yes, sir. How are you? Definitely
Fine.
fine.
How
He
were the elections in Bengal?
Yeah, that is, yeah, you just stole my words instead of giving me opportunity. We are just simply frustrated and like fighting that we should not be allotted the duty which we are sort of a bound and forced rather. So
And
at this point of time, we are bound to do something that we don't know rather. Those trainings are just like trainings, like it is from an administration, I mean, from a total academic to total administration, and that too a risky one. So the
exactly.
concentration in academy is like coming to a negligible amount last until 29th. So, but this topic is too
Lekhna will
much.
tell about the postbiotic and what presently we are doing
Yeah,
in a more briefly.
yeah, like
Lekhna, are you
presently.
there?
Uh,
Yes, sir. Yes, sir. I'm there, sir. So, sir, may I speak, sir?
Yes, Rekana.
Yeah. Very good evening,
Malini
everyone.
madam. Where is Malini madam?
yes, sir.
Yes, madam. You want to share something, madam?
No, no.
Hello.
Actually, I'm not working in that way, but we are just making
Okay.
a few drinks, trying to make people aware, and those that much. Research is
Okay.
not
Thank you
as
so
such
much,
going
madam.
on. Yeah, yeah, yeah. Instead,
Thank you.
I wanted some idea. I mean, to get some more
Lekhna will tell.
thoughts.
She is
Yes,
there
yes,
online.
yes, yes,
Yes,
yes.
yes.
Yes. Very good evening, everyone. Myself, Lekhna, and I'm doing my PhD under Halami, sir, right now. Like we are working mainly on postbiotics. We have I extracted postbiotics from three different probiotic strains, like Lactobacillus fermentum and Bifidobacterium brevi, which was isolated and characterized in our own lab. So from these strains, we have extracted different postbiotics like cell-free supernatant, exopolysaccharides, intracellular proteins, and these things. And we have studied for in vitro anti-diabetic and anti-hyperlipidemic abilities. Like we have tested for amylase inhibitory property, glucosidase inhibitory property, and lipase inhibitory property. And then we have also checked for the short chain fatty acids, which is present in the cell-free supernatant. And based on this, we screened all the postbiotics and selected the cell-free supernatant for in vivo model. And our in vivo model was a type 2 diabetic model. So in type 2 diabetic rats, we administered postbiotics for around 6 weeks. After which now we have seen like a very characteristic improvement in the pancreatic beta cells upon HNE staining, and also the level of insulin produced is also higher in postbiotic treated group. We are yet to perform few experiments to come to the final outcome.
Yeah, but what is the outcome of your results? How the standard drugs work and how the postbiotic showed the activity?
Yes, sir. Like the for standard, we had used metformin and orlistat, which is already there in market for treating diabetes. So compared to diabetic control, our postbiotic worked very similar and in fact better than the standard drug. Like it improved the insulin production in diabetic models, and also the biochemical parameters which we studied like SGOT, SGPT, and the triglyceride level. Also, our postbiotic administration improved the concentration of HDL. Hello.
Hello.
Yeah.
Yes, you are audible, Lekha.
Yes, sir. Yes, sir. So, yeah, in the cholesterol profile, we checked for triglyceride, HDL, LDL, and total cholesterol, where postbiotic administration showed a better improvement in the HDL cholesterol, and it reduced the LDL and triglycerides. So, in liver function test also, the SGOT and SGPT enzymes, and also ALP, there also we found significant results. And kidney function test we are yet to perform.
Okay. Hello.
Good evening. Good
Good
evening,
evening.
everyone. Good evening, everyone. Myself, Pratna Sharma. I am a PhD research scholar from Surat, Gujarat. And I have worked on prebiotics, not postbiotics, but I have worked on prebiotics in my MSc dissertation, in which I tried to isolate and characterize the lactobacillus species from a natural source, which is regionally present in the South Gujarat. Like it's a palm sap, tadi. It is regionally called as tadi or Nero in Gujarat. We actually isolated the probiotic or prebiotic bacteria, lactobacillus, from them, from the palm sap. And we isolated and we found that the species that were found in it were Lactobacillus plantarum.
Okay, thank you, Pratna. So now pursuing your PhD, Pratna.
Yes, sir. My coursework is going on.
Okay.
Yes, sir.
And you started some work on your PhD also?
No, sir. I'm going to start, but I'm trying to make up my mind on a topic related to bacteriocins.
Oh, very good. Bactricin also considered as a postbiotic.
Yes, sir. Yes, sir. That's why I'm inclined towards this topic only. So I'm trying
Exactly.
to make up my mind on this. Yes, sir.
Hello, Ambarish Sindagi.
Who is also available, she can tell her opinion. Mr. Murugan is there.
Yes. Please unmute. Because
Please
this one is the
unmute and tell.
platform where we can connect with each other, we can share our knowledge with each other so that scholars and students get benefit from our knowledge.
Good evening, sir. Good evening to all.
Good
I'm
evening.
traveling in the Bluetooth mode. But however, our lab also works few postbiotics. So, scholar said we also work on the diabetics as well, anti-inflammatory postbiotics. This postbiotics will be derived from few seed, especially Java seed. Um, uh, and also we work on some spices and its fermented product yet to characterize. So we'll be happy to collaborate with the working study as well as the target identifications. We majorly work on AMPK pathway, which uh, works on the hemostasis, especially energy metabolism in the human being. Thank you for this.
Thank you so much, sir.
What is the source of postbiotic you told me? Murugan, sir, what is the source of postbiotic?
Yeah, we have a few kernels. So the kernel found to have a lot of phenolic as well as the bioactive
Yeah,
molecules.
bioactive substances or
Yeah,
phenolic, we can tell them. Postbiotic in general term actually is defined by
No, we metabolize. We
metabolites from bacterial fermentation.
yeah, certainly. See, the phenolic as well as bioactive molecules further metabolized with probiotic bacterium, and we extract and we look for the postbiotics.
Exactly.
Including short chain fatty acids. So yeah, it is under the way. We have found few. Yes, sir. Yes, sir.
నాకు కింద నేను బుడ్డీస్ మెత్తగా వేసుకున్నా కదా.
Someone has to unmute actually.
Recording in progress.
Yes, sir.
Yeah. Network is very poor for me actually today.
So one question from the Ambari Sindagi: can AI application challenges use of postbiotics in future as the therapeutic agent?
Exactly. Yes, yes. Now this is actually the growing area because finally postbiotic also where we have to actually select a large amount of substances. So such dictionary is available with us, right? So we don't know what is the target, where it will hit, what will be the specificity, the dosage. So that actually can be analyzed by AI ML tool, and then you comes to the more detailed bioinformatics interaction studies, and then you test that molecule in a laboratory.
Okay, sir. Thank you, sir.
You can one can minimize.
Yes, sir. Drug designing
Hello.
is also possible.
Yes, yes.
A lot of tools are available to design a drug for a specific target, and we can make even machine learning also for the simulation work. There are a lot there to explore for that. Thank you so much.
Thank you, sir. Sayuri Singh is present. Sayuri, do you want to share something, Sayuri?
Good evening, everyone. I'm a first year master's student. And honestly, I
First,
do not.
introduce you from which institute you are, Sayuri Singh?
Um, I am from Sister Nivedita University, currently
Okay.
pursuing my master's, and I have finished my bachelor's from North Bengal St. Xavier's College. And before I joined this meeting, I was kind of doing some basic studies on postbiotics since this is a new concept to me. And I was reading and I read that postbiotics do have a lot of implications and benefits for various diseases. And I wonder if postbiotics, since I've read that they can influence the gut brain axis, I wonder if postbiotics confer any sort of health benefits like for mental health issues like anxiety, and how much research progress has been done in that area.
Okay, thank you, Sayuri.
Yeah, this is actually very useful and important questions. Now, postbiotics are replacing our all conventional drugs because of
Sir,
nature. Can you hear me?
yes, sir, you are audible.
Yeah, because of the safety issues, and then more importantly, no side effects on this postbiotic. And as already we were discussing that we actually checked the postbiotic for hyperlipidemia and diabetics, and then they work much better. Our preclinical trial result shows than the conventional standard drugs with no side effect. Plus, with reference to curing the disease and with reference to the gut microbiota modulation, they are actually more useful because standard drug basically inhibits the friendly bacteria. So this is what actually one can measure difference can see. Of course, our gut microbiota results has to come in, but then we expect that the where we use the postbiotics, we will have a more population of the scene that gives a better results uh, on the curing the disease as well as on the gut microbiota modulation and then cytokine modulations again, influence on the cytokine also. So such several different advantages postbiotics offers. And then there should not be a very long way to see whether neurodegenerative diseases also can be answered through the postbiotic. Maybe literature is there. And we have actually seen, especially for the autism spectrum disorder. This is also one of the neurodegenerative disease. Postbiotics works almost on a similar fashion like how your probiotic organism works or the gut microbiota works. So very actually useful study. What we need is now very rapidly the preclinical, the dosage, the duration of the study, and how specifically it can be explored. So this is what actually the research is required. Yes. Any other question?
So one basic question, sir, from my side to you and also the Murugan sir, that what drives you to focus on the postbiotic rather than probiotics?
That is question in the chat box.
Not this one is
No,
my
it's
question.
asking. You are asking.
I am asking, sir, not in the chat box.
Tell once again.
Means what drives you to focus on the postbiotics rather than the probiotics?
Oh, exactly. Now, as you told in the introduction itself, once we say the probiotic, then live organism viability is important. Then their transportation, the packaging, storage stability is important, right? And then more importantly, if they have suppose the virulent genes, then we will not be able to. Now you see several of this gram negative bacteria, like today only I was seeing Prevotella, gram negative, bacteroid, gram negative, but then they are actually very useful for a gut health. So I, in fact, in our diabetes meeting, we questioned several of investigators who are using these microorganisms, but then they could answer that question that they are not using as a live form, because when they use live form as a gram negative, then there will be some sort of a virulency. There will be the lipopolysaccharide production, which is actually LPS is a strong. But if you inactivate, then only that cell debris, where the LPS will be inactivated, cells will be dead, and then that can be used as a postbiotic. I hope it is clear. So gram negative,
Yes, sir.
which are potential probiotics, but being they are a pathogenic nature, they cannot be used as a live in the live form for a direct consumption. But once they are converted as a postbiotic, it can be used. I hope now it is clear.
So the next question is how postbiotic
Yeah.
influence the gut microbiome without the living organism.
Yeah, exactly. Because see, finally, this will no the how the probiotics actually no. This is more with relation to the disease prevention, right? More with relation to the immunomodulations. Okay. Like exopolysaccharide is there, EPS is there. More related to the anti-inflammatory markers. For that purpose, it is actually working on that. With relation to the gut microbiota, what happens is they are actually in a very small quantity are effective in killing the pathogenic bacteria, but not the friendly bacteria. So through that, we expect that the postbiotic will be useful even in the gut microbiota modulations. And that is what rightly told initially only. When we use such preparation, it is not inhibiting the friendly bacteria, but it is inhibiting the pathogenic bacteria. And that's why it can be for the gut microbiota modulation. I hope now it is quite clear.
Yes, sir.
Wonderful.
So one more question from Silpa Savalgi. Bacteria from fermentation like lactobacillus
Yes,
can be used as postbiotics.
definitely. You have to actually screen, characterize, fractionate, see the activity, and compare, and then see the active fraction, how increase the active fraction, and then that active fraction has to be further characterized by LCMS or HRMS. And then you see in the dictionary that whether such molecule has been characterized before or not. So such postbiotic based drugs are available now in the commercially available in the market. In fact, last gut microbiota conference, I was one of the panelists. This year they did not invite me.
Okay, fine. Think that Silpa got her answer. So
For
one more
the
question.
yes.
One more question from the Ambaris. Any
Uh,
modulation through AI
yes.
in postbiotics which helps to improvise gut microbiome of immunologically challenged people?
Yes, yes, possible. Because these are actually the extract from the friendly bacteria. These are actually the extract from the health beneficial bacteria, metabolites, right? So for that, actually, our immune system or the gut microflora recognizes. They don't get inhibited with that, you know. Just like eating a curd. So whatever this acidic pH is there, lactic acid is there, it do not inhibit the friendly bacteria. But if it is a pathogenic bacteria, will not allow it to grow, will not allow to multiply. And then this is how actually it is being used or useful.
So, sir, the next question, Samay. So, these postbiotics are the safer alternatives for the immunocompromised patients compared to the probiotics. Hello.
Yeah, you have to repeat.
Means these postbiotics are safer alternatives for the immunocompromised patient compared to the
Yes,
probiotics.
exactly. Very, very correct and very logical question actually. Like E. coli nasal strain, we suppose not to take as a probiotics, in other words. Okay. Previotola, we suppose not to take. Bacteroid, we suppose not to take in the form of probiotics. But in the form of postbiotic, yes. You know, just Agarkar Research Institute director itself is, I had interaction with him. They are defended their project, and then I have to say yes. Why? Because their immunogenicity or pathogenicity we are inactivating another useful fraction which is required for immunomodulation. Why E. coli nasal strain actually is considered as a probiotic because of the very good immunomodulation, right? But E. coli being a gram negative and lipopolysaccharide production ability, we can't explore as a live bacteria as a probiotics. In other words, I hope I
Yes,
have.
sir. One more.
Please.
Hello.
Yes.
Thank you so much, sir. So one more question from Rabi, and I would like to hear this from the Prarthana and Tejaswani. That what is the role of synbiotics? Can you brief on it, sir? Prarthana and Tejaswani, are you present there? Prarthana
Yes,
and
sir.
Tejaswani.
Yes, sir.
Okay, so one question.
Yeah.
Network
Yes.
is very poor actually.
Yeah. What is the role of synbiotics? Can you brief on it?
Yes, sir. A synbiotic is nothing but the prebiotic and the probiotics. The prebiotics will just in acting as a food for probiotics, and then whatever we are getting the postbiotics, we'll say it is a synbiotic. It is in a mutual this thing. If it is comes from the prebiotic and probiotic, we will have a synergetic effect on the postbiotics.
Thank you
For
so much,
example,
Tejaswani.
yeah,
Yes,
yeah.
Tejaswani. Please,
For
please go ahead.
example, I would like to give is a phytochemical is a prebiotic, and the probiotic we can take like lacto and bifido, and if we ferment together and it releases the same postbiotic, so we will say that the synergetic effect is bidirectional from prebiotic to probiotic and probiotic to prebiotic. So there is an synergetic effect. So it is nothing but a symbiotic.
Okay. Thank you, Tejaswani.
Yeah.
Ravi, I think that I got your answer. Ravi?
Question.
Yes, Sairi.
Any other question?
Yes, sir. Sir, because of this particular development, is it possible that we could possibly reap the benefits from those bacteria that are too weak to survive in our gut, and function as probiotics, since like the postbiotics don't need to be alive, so that can we use those bacteria that are too fragile to function as probiotics and use them as postbiotics?
Yes, yes, it is a valid question. I already explained, I believe. This is as the advantages of the postbiotic over probiotics.
Okay, sir. Okay, sir.
Sir, one more question from the Ambarish Sindagi. How far good the use of postbiotics for children of 1 to 10 age?
Yeah, actually, for the children, as such, probiotics are no recommended, but then with the care, right? Because there can be a immunological reaction to some extent. But if we see the Panigree's paper, the newly born children, they have given lactobacillus. And then they could actually see the prevention of sepsis. That paper is in Science
Okay.
Study in Orissa. The team from US in collaboration with AIMSA, they did, for children. So postbiotic also can be consumed, but otherwise also, in most of this our fermented product, which are otherwise also cooked or baked. So what we consume is the postbiotic. You come to South India, whatever the idli or dosa you eat is a postbiotic, in other words. You will see even in pickle also, after some days the microorganisms will be dead. So you have actually the postbiotic here. So several such I think product you will see everywhere, where we after fermentation we cook, or then you just see these fermented vegetables like this Sikkim. We have the gundruk, khalpi from
Yes, sir. Kundra.
then sinki, radish. So you ferment and then you sun dry, right? With those microorganisms. Or then you have even the fermented bamboo shoots. So we don't eat that one as such, but then we cook or fry at least. So that means we are converting those live organisms and their metabolites for this useful purpose. So in other words, we are actually consuming the post.
So you're not audible now, sir.
Do the comparative studies, identify the dosage here, and then see the disease curing ability. Then you actually are coming up with a new functional molecule for the disease prevention or curing or disease management. And this is actually a very important topic because our source is the microorganisms, right? Where most of you people are working on one other microorganisms. So your expertise or today's lecture will be actually starting material for our career in future, our startup in future, or the to start the entrepreneurship also.
Yes, sir. Related to your this entrepreneurship, one more question from the Anjali Jaglan.
That's
What
your
are the biggest challenges?
Hmm.
Yes, sir. One more question is, what are the biggest challenges in translating these findings into real world human use? And what delivery formats such as the functional food, capsules, or pharmaceuticals are likely to be most effective? Hello.
Hello? I couldn't hear fully the question, but I think she herself can also answer this question, or she can tell at least her opinion.
Anjali, are you there, Anjali?
Yes,
Dr.
sir. Yes,
Anjali?
sir. Yes, sir. I'm here, sir. Yeah, sir, I think we can use in functional foods, but I was thinking about how can we, like, if we are giving in capsule format, can we
Yes,
use the postbiotics in capsule format also, like
yes, yes, yes.
sachets or capsules? Because in functional foods and other food, we can incorporate it. just like dosa and idli you were talking about. Those are functional food. So we can also give them in capsule format, I guess. So we,
Exactly, whatever are there commercially available, they are in the capsule format.
yes, sir, yes, sir.
But now,
Ye-
as a functional food, you incorporate, convert it in the food product, and then you can actually consume regularly. So
Okay,
this is
sir.
what actually
Okay.
this functional food concept is for
Some
this.
yogurt or lassi and all, like dairy products can be made, I guess,
Yeah,
from it. Yes, sir. Okay, sir. It was very good, sir.
exactly.
Hello. Thank you. Thank you, Anil. Yes, yes.
Tiwari we can conclude. Now almost close to 9.
Yes, sir. Just one question is there, sir.
Okay.
Means from the Ravi and the
Yeah.
Porna. From the Ravi, which one is the best probiotic strain, sir? There are several strains available in market. How can it can be categorized?
Tejaswini will answer this question.
Yes, sir.
Tejaswini?
Actually, yes, sir. In market, available is VSL 3, BB 212. All those strains has their particular own functionality based on their characterized attributes of all GI and everything. What we are targeting is that when we incorporate it in the product, we are seeing the viability of the count of the bacteria, which when we consume, at least we should reach 10 power 6, 10 power 9 billion cells of CFU. But many of the market which is available of the product, it is almost when we test is like it not reaches. And even the product also they have mentioned 10 power 9 billion cells. If it is targeting that, it would be better compared to the viability or consumption of functionality into the gut is much more we will characterize into that. Especially the bifido and everything is as we know that's an oxygen, that's it's an anaerobic. So probably we will check on to the viability, and then we'll characterize into which one would be a better in taking through the food or incorporated into the products.
Okay. Thank you, Tejaswini. One more question for you.
Yeah.
Is
Yes,
there
sir.
a mechanism that the gut favor the good bacteria over the bad, and during or when there is an infection caused by food or waterborne contamination?
Mhm. Yes, sir. Actually, it is previously all the probiotic strains which we are like targeting is almost it'll enhances the gut barrier infection, immunomodulation, as sir has mentioned, cytokine profiles and everything. As we get the waterborne infections or something, the primary antibodies or the secondary antibodies which is released in the blood. So if you're taking the probiotics, it is acting as an anti-inflammatory, which is against it'll act, and so it has many properties which enhances the gut health and keeping the mucus layer also to inhibits the pathogen. So one basic question is that probiotic increases the IgA production, that is IgA, it's immunomodulation, which increases the mucin level so that the pathogen will not enter into your body, that will not colonize as such. It inhibits the pathogen as a competitive exclusion principle. So this is how the mechanism goes with NF kappa B pathway.
Thank you so much, Tejaswini.
Yes, sir. Thank you.
I hope that all the participants got the result. So thank you everyone for joining today's session. My special thanks to Dr. Prakash Salami sir, Dr. Murugan sir, Malini madam, Dr. Anjali, Dr. Tejaswani, and Sayuri Singh for their contribution and sharing their research insights. So thank you everyone for joining today's session. And also I have shared one feedback form. Okay? So just fill out that feedback form so that we can send your e-certificate to your mail ID. Once
Thank
again,
you,
thank you.
Bibrang sir. We want more of this type related to this topic only, and with Halami sir, because we are like
Okay, sir.
poor little lab sitting at one corner of West Bengal. So like it, especially the entrepreneurship related matters, something we want to
We
hear
may
from
continue
you
this
later
topic on the
on.
next
Yes,
week
yes,
also,
sir.
ma'am.
Yes, sir. Definitely.
Yeah,
Thank
we
you.
can refine little bit and then we can come back again on the similar line.
Yes,
Sure,
sir.
sure, sir.
Sure, sir. Yes.
Thank you, everyone.
Thank you, everyone, for joining today's session.
Thank you.
Thank you, sir.
Thank you, Silva.
Thank you very much, sir. Thank you very much.
Thank you.
Thank you, sir.
Thank you, Saidi.
Thank you, sir.
Thank you.